Yes, I see that your screen is now shared. It's not yet. Yes, all good. Thank you very much. And by the way, good morning.
Good morning. Good afternoon.
Thanks.
Good evening, actually. Okay.
Well, yeah, kind of afternoon. Thank you very much. And so, yeah, Tandy, the floor is yours.
Thanks. So, Christophe, thanks for that talk, because what you talked about is actually related to what I'm going to be talking about. So I really appreciate it. So I'm going to be talking about some new work in getting species tree estimation in the presence of gene duplication and loss. So everyone knows what a species tree is, so we don't need to go into that, but we are talking about species tree estimation, not gene tree estimation. And we're talking about using genome scale data to do this. And we're going to call that phylogenomics. So the standard way one talks about phylogeny estimation is you are taking sequences and you're trying to infer the tree. And I'll just note that this tree that you're inferring is not rooted. And that's because the models of sequence evolution are time reversible. So it's really easy to see that if you have very short sequences, you don't have really a chance of getting the true tree. But that as the sequence lengths increase, error should go to 0. At least you would hope they would go to 0. And that leads to the question of, is a method statistically consistent under a given model of sequence evolution, such as under Jukes-Cantor, under the GTR model? And so what you're saying there is, as the sequence length increases to infinity, can you prove that the error will go to 0? And I just want to note that this is something you cannot demonstrate with a simulation. You have to have a mathematical proof. So statistical consistency has been considered a desirable property for phylogeny estimation, and a lot of work goes into making sure that the methods that are used are statistically consistent. And if they're not, then understanding the conditions under which they're not. So genome scale data gives you this idea that maybe you can get at the species tree because you have enough data, maybe, okay? The problem is that across the genome, you can have different trees, and there's multiple causes for this. We actually heard about 3 in the previous talk. Incomplete lineage sorting is one, gene duplication and loss is another, and horizontal gene transfer is a third. All of them can lead to differences in the gene trees as you move across the genome, and they all can cause problems. The one that's the most well understood is incomplete lineage sorting, so I will spend some time talking about that. But gene duplication and loss is what leads to paralogy, and I will talk about that as well. I won't actually talk about horizontal gene transfer today. So I just want you to note that if the true species tree has human and chimp as siblings, then you would have it over here. But over here, the gene tree has human and gorilla as siblings. So there can be different gene trees that have different tree topologies. Okay, so this kind of problem shows up in biological data sets. This is just one example from the 1,000 Plant Transcriptome Project. In 2014, we had a study in PNAS. In 2019, we had another study in Nature. And in both of those cases, there were multi-copy genes. So multi-copy genes is what you get with gene duplication and loss. And in this particular study, we had to reduce from 9,500 gene families to 400 because we could only work with the single copy genes. So that's just an indication of how common gene duplication is. And the other thing that we noticed was massive gene tree heterogeneity that was consistent with incomplete lineage sorting. So both of those things were showing up. Again, this is the point that the first thing is evidence of gene duplication and loss. And the second one is evidence of incomplete lineage sorting. So when you think about developing methods for species tree estimation, you need to think about developing methods that are actually robust to multiple causes for discord. So today's talk will be a brief introduction to how to do species tree estimation when you have incomplete lineage sorting. And I'll be focusing on 2 methods, one which is pretty well known, which is called Astral, and the other one which is not very well known at all, called Astrid. And then I will talk about a breakthrough in the last like 1.5 years, a sequence of breakthroughs for species tree estimation in the presence of gene duplication and loss. And the first breakthrough was a proof that Astral Multi, which is a version of Astral designed for multiple individuals. Now, remember, Astral only deals with incomplete lineage sorting. So there was a proof that that version of Astral is statistically consistent for GDL. That was the first breakthrough. Then there was a modification to Astral to make it work really well for gene duplication and loss, and that's called Astral Pro. Then there was another method, Astrid Multi, which is like Astral Multi, but designed for multiple individuals. It's also really accurate. And then Fast Muller FS, which is a new method that was developed specifically for GDL, and it has also excellent accuracy. So I'm going to show you results comparing these methods on simulated data, okay? So I'm going to start by talking about incomplete lineage sorting. So remember this thing about gene tree to score due to incomplete lineage sorting? This actually happens specifically with rapid radiations, and human, chimp, gorilla, and orangutan is one example where it happened. You have a hierarchical model of sequence evolution. So what you need to think about is that instead of having one tree on which all the sequences evolve, the species tree generates gene trees. The gene trees don't have to be equal to the species tree. And they're being generated for ILS, they're being generated under the multi-species coalescent. So you have gene trees that are different across the genome. Then each gene tree evolves sequences. And that can be under Jukes Cantor. In this case, I'm showing GTR. Okay, in fact, I'm showing GTR with indels since there are dashes. But think of MSC to get gene trees, GTR to get sequences. So how can we estimate species trees into this hierarchical model? The most common thing is just to do concatenation. And of course, concatenation means you put all the multiple sequence alignments together and you run your preferred maximum likelihood heuristic, okay? So that's one approach. Another approach is to what we call summary methods. These are methods that estimate gene trees and then combine the gene trees. Then there are methods that are called site-based methods where you bypass the gene tree estimation entirely. It's not though, it's like a concatenation analysis, but it's not a standard concatenation analysis. You're basically looking at the site patterns in the combined alignment and using that to directly get the tree under the MSC plus GTR model. And the most well-known of these methods is SVD quartets. And it computes quartet trees and then you combine the quartet trees. It's very computationally intensive when you have large numbers of species. Then there's co-estimation methods like StarBeast and StarBeast2 that co-estimate the gene trees and the species tree together, even more computationally intensive. So of these four types of methods, only the top two are used on large data sets, and they're like dominant in practice. So let's just talk about concatenation. If you concatenate all of the alignments together and then just run maximum likelihood, it's not statistically consistent if you have incomplete lineage sorting. And that was a mathematical proof by Sebastian Rock and Mike Steele. And worse than being inconsistent, it actually can be positively misleading, which means that as you add more and more genes, you can converge to the wrong tree with probability going to 1. And what that means is that your bootstrap support is going to be 100% for the wrong tree. Not a good situation. That's theory. In practice, it's actually sometimes the right thing to do because sometimes the conditions actually are better for a concatenation analysis than for any other alternative, current alternative. So simulations show that it's not so clear-cut. Okay, let's go back to this hierarchical model. Remember, the species tree generates gene trees, the gene trees generate sequences. What you'll notice is that there are different genes are not evolving down the same tree, right? Which is why that combined analysis, concatenation analysis doesn't really work because it's assuming everything's going down a single tree. Okay, but if you think about this hierarchical model, you can try to invert it. So you can go from the gene sequence alignments and try to estimate gene trees. And once you have the estimated gene trees, you can use the probability distribution under the model for gene tree topologies and try to infer the species tree. And that's exactly what a summary method does. It reverses this process. It takes the alignments and gets gene trees, it takes the gene trees and gets the species tree. One of the things I want to point out here is that getting gene trees on different data sets is actually a lot faster than doing a concatenation analysis because the number of sites is so much less. So this is actually faster than the concatenation analysis and it's easily parallelized. Okay, and it can be statistically consistent if done properly. So Astral is the most well-known of these statistically consistent methods. It was developed initially in 2014, but it's evolved over the years. It has lots of different bells and whistles now. It's very, very, very popular, very fast, very accurate. Astrid is one that people know a lot less about. So Astrid takes the same input as Astral, but then it computes a matrix of leaf-to-leaf distances. And that matrix of leaf-to-leaf distances can then be passed to a distance method like neighbor joining, but in this case, we use FastME, which actually has better accuracy than neighbor joining and is actually faster. It's also statistically consistent under the multi-species coalescent. It's polynomial time. And what's nice to know is that it's relatively as accurate as Astral. There's some conditions where it's more accurate, some conditions where it's less. And we don't really understand the conditions under which you would prefer one over the other, but it's faster. Okay, so what I've shown you are Astral and Astrid. These are two types of summary methods. And just to remind you, Astral operates by computing quartet trees. Basically, it takes the gene trees and it represents every gene tree by a set of quartet trees, and it uses that quartet tree representation to infer the species tree. Astrid operates by taking the gene trees and getting a distance matrix. So these are very different ways of getting at the species tree. Okay, now I'm going to switch. Okay, I'm going to switch over to gene duplication and loss, because that's the topic we're covering here. I want you to see what a gene family tree can look like. So if you have a single duplication at the root of a species tree, in this case, it's a rooted species tree, you can get 2 copies of the species tree in your gene family tree, okay? So your gene family tree has extra copies of the species. And that makes inferring the species tree kind of complicated. Now, in this particular case, it's easy, right? You can just say, okay, I've got 2 copies of the species tree. That's what I want. But you can have more complicated situations with losses. And if you have duplications and losses, you can get to all sorts of stuff. So in particular, on the top left, you have the species tree right here. This is one gene family tree, which has 2 copies of the species tree on ABC. This is also a gene family tree that has lost 1 copy of the B species and 1 copy of the C species. But look at this one, the one in D. It's lost a copy of B, it's lost a copy of A, it's lost a copy of C. So this is a single copy gene tree. It doesn't have any duplicate copies, but it is not the same as the species tree. So you can get single copy gene family trees that are not identical to the species tree. And that is actually problematic because you might think it should be the species tree, but it's not. Okay, so single copy gene family trees don't have to agree with the species tree. Losses can really complicate stuff. Okay, so under gene duplication loss, just like under incomplete lineage sorting, there are these different categories of methods: concatenation, summary methods, and coestimation. Like ILS, coestimation is too expensive, so people don't use it. Summary methods do exist. The most common ones are like gene tree parsimony approaches, but there's also supertree methods. And then there's a Bayesian method that's been developed as well. I will talk a little bit about them in a second, but concatenation is actually really interesting to talk about. If you have duplications, how do you do concatenation? There's 2 ways. One is to figure out orthology. And if your orthology is perfect, you can do this. And if your orthology isn't perfect, you can still reduce to single copy stuff, or at least groups of orthologs. You can do concatenation, okay? The problem is that orthology isn't that reliably done yet. So concatenation relying upon orthology can be a problem because orthology isn't solved. The other approach is restricting yourself to the single copy genes. Okay? If you restrict yourself to single copy genes, you're throwing out data. So concatenation right now has relied upon one of those 2 techniques, orthology or reliance on the single copy genes. And just to remind you, in that 1KP analysis, when we restricted ourselves to the single copy genes, we threw out more than 9,000 of the 9,500 genes. We threw out most. Okay, so let's talk about summary methods. Here, the input is a set of gene family trees, and you want to estimate the species tree. You don't have to know orthology, and you don't have to throw out anything. You just take all of your gene family trees. I will note you have to get the trees though. So you do have to deal with getting trees on each of your gene sequence alignments, and that is not a trivial problem. But let's suppose that we've done this gene family tree estimation for all of our genes. The first proof that anything in this area worked that was statistically consistent came out in 2019 and then published in a journal in 2020. by Sebastian Rock and his student Ben Legreed, as well as Erin Malloy and myself. And it proved that astral multi was statistically consistent. Remember, astral is designed for ILS, but the multi-individual version was provably statistically consistent under gene duplication and loss and runs in polynomial time. First proof. Second theorem, just a few months later, was that a supertree method called fast mul RFS, which is a supertree method designed for mul trees, is also statistically consistent, but not under all GDL models, but under a subset of the models where there's no what we would call adversarial gene duplication and loss. And examples of that are duplication only and loss only models. So two methods that can be used with gene duplication and loss that run in polynomial time. The first is provably statistically consistent throughout, and the other is only under some conditions. The big breakthrough was Astral Pro. So Astral Pro was a redesign of Astral in order to make it work specifically for gene duplication and loss. And the design is that, remember, Astral encodes all of its quartet trees, all of its gene trees as quartet trees. So what was done here is that all of the gene trees are rooted and then tagged. Tagging means you identify each node as either a duplication node or a speciation node. And once you've done that, then you can modify how you encode your quartet trees, your gene trees as quartet trees, and only use the ones that are actually going to be representative of speciation. Okay? So you get these quartet representations by doing the rooting and then the tagging. Astral Pro. So why was Astral Pro a big breakthrough? Because when Astral Pro was compared to Astral Multi, it was much better. And Astral Pro was also compared to other methods and was much better. But it was not compared to fast mul RFS, and it was not compared to Astrid Multi, which is a variant of Astrid for multi-individuals, okay? So that's what my students have just done. This is unpublished work now. We looked at data sets with 100 to 1,000 species, up to 10,000 genes. Every gene evolves with gene duplication and loss and incomplete lineage sorting. And we vary the sequence length per gene so we can look at gene tree estimation error and see how it impacts things. And we compare Astral Pro, Astrid Multi, and fast mul RFS. And I just want to remind you that none of these are actually proven statistically consistent under gene duplication and loss, but all of them have been shown to be better than Astral Multi, except that Astrid Multi hasn't been compared to Astral Multi. Okay, so we did this comparison and we report species tree error, which is the standard bipartition distance, normalized to be between 0 and 1, also called Robinson folds. And we also look at the running time, but this is running time after the gene trees are estimated. Okay, so I'm just going to show you a sample of the results. This is what happens as you increase the number of gene trees. These are 100 species data sets. And what I'm going to show you is that the smallest number of genes is 100. And with 100 genes, which is right here, the pink is fast mul RFS, blue is Astral Pro, green is Astrid Multi. Fast mul RFS is not quite as good. This is about 12% error in the species tree. These are both like 7 and 8% error, okay? So at 100 genes only, there's an advantage to using Astral Pro or Astrid Multi. As you increase the number of genes, all the methods look about the same, right? And the errors drop. Okay, so increasing the number of genes improves accuracy and the differences between methods disappear. Now, in terms of running time, again, this is Astrid Multi in green. Astrid Multi is the fastest. Fast mul RFS is the slowest. So that's the first experiment. Second experiment is the impact of incomplete lineage sorting. So I want you to remember that there's duplications, losses, and incomplete lineage sorting going on. Plus we're dealing with estimation error in the gene trees in all these cases. So this number 0% says that there's no ILS. This is 20% is a moderate ILS, 50% is high ILS. So what do these numbers refer to? It's the distance between true gene trees and true species trees measured using Robinson folds. So this is saying the 20% case says the true gene trees are different from the true species trees in 20% of their edges. 50% says that the true gene trees are different from the true species trees in 50% of their edges. High ILS, moderate ILS, no ILS. And what you can see is under all three conditions, under these two conditions, the methods are about the same, right? No real important distance. Here, under the high ILS, fast MOLR-FS is worse. Okay, running time, Astrid multi is the best. Fast MOLR-FS is the slowest, Astral Pro is in the middle. Okay, now on the big data sets, 1,000 taxa species trees with 1,000 genes. And these are short sequences. So there's the gene tree estimation error is about 44%. And there's moderate ILS. You can see all the methods are about the same. They're getting only 5% error, okay? 5 or less, 3% error here. So they're all about the same. The running time does distinguish the methods, with fast MOLR-FS being the slowest, Astrid multi being the fastest. Remember, this is unpublished work, but you can think of 3 different methods you could be working with. So here are the conclusions. You can estimate species trees using all of your data. You don't have to restrict yourself to single copy gene trees. You do not need to figure out orthology. All of these methods work well. Astral Pro and Astrid multi are a bit more robust than fast MOLR-FS. All of the methods are actually pretty fast. Even in the biggest data set, they all finished in under 7 hours, which says you wouldn't need to do 1,000 species by 1,000 genes. And mind you, with multiple genes, you know, this could be a very large data set. You don't have to do it. You can just get the gene trees and combine them, and it's actually very fast. Statistical consistency has been established for Astral multi, but not for the other methods, but the other methods are more accurate. So maybe some of them actually are statistically consistent. We just need proofs. And there's still much room for improvement in accuracy. These are just like the first set of methods that have really been shown to work well, and there will be better ones coming, okay? Even on large data sets. So with that, I'll just thank the people who do the work. Siavish Mirarab, former student, is the developer of Astral. Erin Malloy, also former student, developed fast MOLR-FS. And James and Rinmoy are the 2 new students who are working on this comparison between existing methods. And I can be reached by email if you want to follow up. And also I posted this talk on my website, and I put the link for it at the top. So I guess I'll take questions now.
Okay, so thank you very much, Tandy, for this great talk. So indeed, we welcome questions. Feel free to ask them either in the chat or by speaking up. I see that there is a question in the chat already. So Susanna is asking, for a within species phylogeny inference, which method would you suggest as the best method?
Wait a minute, which is the best method for just species estimation?
Yeah, within species phylogeny inference.
This is not gene tree estimation, this is species tree estimation?
Susanna, would you like to speak up maybe to specify?
Within species, you're saying when you have a single species. Yes, is that what you're saying?
I think so.
Oh God.
Yes, exactly.
I'm sorry, I actually don't have an answer for that one. I really don't. So, you know, I actually find everything about doing things within a species kind of problematic, because, you know, you're not inheriting typically from just one parent. And if you're not inheriting from one parent, I'm not even sure what a phylogeny represents. But if you're always inheriting from one parent, then it's just the same stuff, I would say, except that, you know, you have less change. So I would say it would be important to really use something that is able to deal with very slowly evolving sequences. That's what I would say. But I'm sorry, I don't really have a good answer to you. This is sort of out of my comfort zone. So.
Okay. No, because I used so well, maybe I can email you later.
Yes, please. And we can have another Zoom chat sometime.
Yes, that would be great. Okay, thank you.
Sure.
Thank you. Okay, so feel free to speak up if you have an extra question for Tandy. Actually, I mean, I have one. And so about this simulated data that you use for comparing the methods to one another, can you say a bit more about how you simulate the data, like how you make it?
So we use SimPhi. We use SimPhi for the species to generate gene trees from species trees.
Yes.
And then we use Indelible to generate sequences down the gene trees. And these are data sets that so then we get the true alignments. We didn't estimate alignments, but then we take fast tree. So this is answering Luizas. We used fast tree on the alignments.
Okay.
So what's interesting is that fast tree, a lot of people would prefer not to use fast tree and they would prefer to use Raxamel. But what's interesting is that in many, many, many conditions, fast tree is a very good method, just as good as Raxamel for tree topologies. So the answer is indelible and SimFi together to get the simulations. And then from the true alignments, we ran fast tree.
Okay, thank you very much. Yeah, you already answered Louisa's question, I believe, right?
And Christophe has a question.
Christophe has a question. So Christophe, yeah.
I've got tons, but I don't want to take up too much space. But yeah, thanks, Tandy. It was really wonderful. And damn you to have like the last word on the use of the importance of orthology or not. But I just wanted to ask about the tree. I mean, how do you then do the routing on the
we're not routing anything. So when we run okay, Astral Pro does routing. How it does its routing, I am not 100% sure, but I think what it does is it is taking each gene tree and trying to route it to minimize the total number of duplications and losses, I think. Now, if you read the Astral Pro paper, it probably says exactly what it does. But Astral Pro is the only one of the methods we studied that actually routes the gene trees, okay? Both fast small RFS and Astrid Multi do not route the trees. They just take unrooted gene family trees as input and they operate from that. So, you know, the key to Astral Pro doing well is its ability to do the routing and to do the tagging. And what's interesting is that it does such a good job, despite the fact that I think these are both hard problems.
So and Florence, you should cut me when it's too much.
It's okay.
So one thing that is really interesting, and I've seen also some other works suggesting also what you observe, is that the ILS models are surprisingly robust to having paralogs in your data set. And I think the intuition, I think, for that is that they don't put a lot of weight in these long distance relationships, right? I mean, what the information, the ILS that is really crucial is when you have, for instance, if we think of human, chimp, gorilla, is the fact that the divergence between human and chimp is smaller, and therefore we will see an excess of really close trees between. But the fact that you see also occasionally some very distant human and chimp, that's not giving you so much information. And I got the feeling that that's a little bit what happens with paralogs too, right? It looks a bit like this deep.
So let me just tell you a theorem that is in all of these papers. There's a theorem that basically says that the reason Astral is correct, is statistically consistent, is that under ILS, the most probable quartet tree is the species tree. The same theorem was proven about duplication and loss and about HGT, random HGT. So there's this interesting thing that is connecting Astral specifically, quartet-based methods specifically. When you encode the gene trees as quartet trees, the basic theorem that makes them work is this idea that the most probable quartet tree is the species tree. That's true under all three situations. So that's this mathematical thing that makes quartet encodings of gene trees a good thing. And this is just a paper, this is I think I'm still sharing. This is a figure that shows when you have ILS and HGT, but you make more and more HGT, at the extreme level of HGT, the method that's the best is Astral. And the method that's the worst is concatenation using maximum likelihood. The second best method is quartet max cut. So there's something about quartet-based encodings of gene trees. This is not the same as estimate quartet trees independently. It's encode your gene trees as quartet trees. So that's this interesting connection among all of these models. Good? Okay, my time is up, but Christophe, you and I can talk again some other time, right?
Yeah, I was going to say, so yeah, thanks again for Tandy for the great talk, and thanks everybody for the discussion.
Thanks for the invitation.
Oh, you're welcome. And I see that we have the extra questions will be moved to the break, sorry.